This is SARS-CoV-2. Like most viruses, the disease it causes goes by a dissimilar name. In this case, the severe acute respiratory syndrome coronavirus 2 is the virus responsible for the 2019 novel coronavirus disease, or COVID-19. SARS-CoV-2 is a member of a larger family of coronaviruses that can cause illnesses that range wildly in severity. These viruses are named for the crown-like spikes on their surface, and they generally circulate among a handful of animals, though they have evolved to infect humans. The first human coronaviruses were first identified in the mid-1960s, though it would not be until 2003 for the first known severe illness caused by a coronavirus to emerge, the Severe Acute Respiratory Syndrome, or SARS epidemic in China. A second outbreak of severe illness would also occur in 2012 in Saudi Arabia with the Middle East Respiratory Syndrome, or MERS. SARS-CoV-2 is a particularly dangerous strain of coronavirus because of its perfect balance of contagiousness, incubation period, and fatality rate. But where did it come from, and how did it become such an effective pathogen? Wei Guxian of Wuhan, China began to feel ill, becoming one of the earliest known cases of a SARS-CoV-2 infection. 6 days later, she would be admitted to Wuhan Central Hospital with an infection in both lungs that was resistant to anti-flu drugs. Staff later learned she worked at a wildlife market later found to be connected to the outbreak. By the 27th, Wuhan health officials were informed that a new strain of coronavirus was behind this new illness. In the coming days, both Ai Fen, a top director at Wuhan Central Hospital, and Li Wenliang, a Wuhan doctor, had posted information on the Chinese messaging platform WeChat about this new virus. Both were reprimanded for doing so, with Dr. Wenliang even being called in for questioning. By December 31st, Wuhan health officials had confirmed 27 cases of the new illness and subsequently closed the market they thought was related to the virus's outbreak. Chinese authorities would officially alert the World Health Organization of the novel strain of coronavirus and the severe illness it causes. As the world was first being made aware of this mysterious new illness, Wuhan Public Security Bureau would bring in for questioning 8 doctors who had posted information about the illness on WeChat. Officials at the Hubei Provincial Health Commission ordered labs which had already determined the novel virus was similar to SARS to stop testing samples and destroy existing samples. Chinese authorities would even report to the World Health Organization that it had seen no clear evidence of human-to-human transmission of the novel coronavirus. Despite attempts to further downplay the impact of this new disease, now called coronavirus disease or COVID-19, the illness would soon start appearing internationally within days. The reality of the contagious nature of SARS-CoV-2 would soon become dangerously apparent. Shortly after the epidemic began, Chinese scientists began sequencing the genome of SARS-CoV-2. Though this was completed by January 2, 2020, the data would not be made available to researchers worldwide until January 9th. This genomic sequence data would unveil the epidemic, revealing that the number of COVID-19 cases had been increasing due to human-to-human transmission after a single-point introduction into the human population. In order to determine the nature of SARS-CoV-2's origin and how it evolved, a collaboration between several research institutions began analyzing several key characteristics of the virus. Among these, the genetic template for the virus's spike proteins that formed its corona was the primary focus. Structurally, SARS-CoV-2 is composed of a spherical shell made up of a lipid membrane encompassing a core that contains the virus's genetic material. As with all viruses, in order to replicate, it first needs to infect a living cell. Protruding from the virus are structures known as spike proteins. Spike proteins are essential for the virus to both attach to and fuse with a host cell, infecting it. The spike protein of SARS-CoV-2 possesses 2 fundamental domains: a receptor binding domain that allows the virus to attach to a host cell, and a second domain which contains sequences that mediate fusion of the viral and the host cell membranes. During infection, the spike proteins act as a key, binding to receptors on human cells. A fusion process is then activated by enzymes present on the host cell. Known as a cleavage site, this enzyme-mediated process unlocks the host cell and allows the virus to gain entry, where it then hijacks the cell's machinery to replicate. While spike proteins are a characteristic of all coronaviruses, the genomic data had revealed a peculiarity of SARS-CoV-2. It binds and fuses to the host cell wall as a result of a cleavage site that's activated by the enzyme furin. The significance of this finding is that furin is not only present on lung cells, but also in the tissues of the liver and small intestine, allowing SARS-CoV-2 to have multiple potential infection points in the human body. However, it should be noted that the exact molecular pathway that occurs during viral entry has not been mapped, nor is the correlation between the virus's activation site and the severity of an infection understood. The nature of how SARS-CoV-2 spike proteins behave also offer a glimpse into its origin. Researchers have found that the receptor binding domain portion of the SARS-CoV-2 spike protein had evolved to effectively target proteins expressed by the ACE2 gene. Expression of this gene suggests that it may play a role in the regulation of cardiovascular and renal function. SARS-CoV-2 spike proteins are so effective at binding to human cells that scientists have concluded that this feature was the result of natural selection and not the product of genetic engineering. The evidence for natural evolution has been supported by data on SARS-CoV-2's overall molecular structure or its backbone. An engineered pathogen would most likely be constructed from a virus already known to cause illness, but SARS-CoV-2 appeared to be substantially different in structure from existing known human coronaviruses. However, it did bear resemblance to several similar viruses found both in bats and pangolins, an armadillo-like mammal found in Africa and Asia. From the genomic sequence analysis, researchers have concluded one of two likely possible scenarios for SARS-CoV-2. In the first scenario, it's theorized that the virus evolved through natural selection in an animal host, then migrated to humans. Strong evidence for this lie in previous coronavirus outbreaks. The 2003 SARS outbreak was attributed to direct human exposure to civets, while the 2012 MERS outbreak was a result of exposure to camels. In this theory, the originating animal of SARS-CoV-2 is proposed to be bats, since its structure is very similar to bat coronavirus. However, due to the lack of evidence for direct bat-to-human transmission, it's speculated that an intermediate host animal could have facilitated the pathogen's movement from bats to humans. Because the distinct features of SARS-CoV-2 spike protein would have already evolved prior to entering humans, in this theory, the current pandemic would have emerged rapidly from either a small infected group or even a single infected individual. In the alternate theorized scenario, the virus jumped from an animal host into a human in a benign state, then evolved to its current pathogenic form within the human population. This theory is supported by the similarity between the receptor binding domain of SARS-CoV-2 spike protein and that of some coronaviruses found in pangolins. As in the case of the first theory, this transmittal may have occurred indirectly through an intermediate host animal. Once within the human population, the virus could have evolved its cleavage site to activate by furin, possibly via limited undetected circulation in the human population prior to the beginning of the initial epidemic. The SARS-CoV-2 cleavage sites appear to be similar to that of strains of bird flu that have been shown to transmit easily between people. This cleavage site evolution could have triggered the current pandemic, as it now made the virus far more contagious. As of April 2020, the SARS-CoV-2 spike protein is being heavily used as a target antigen in vaccine development. The virus's ability to infect human cells so effectively has also made its behavior a curiosity to researchers. However, understanding the spike protein's role in the context of the COVID-19 pandemic is not an easy undertaking, considering the unpredictable nature of coronaviruses in general. It's hopeful that SARS-CoV-2's strength, its spike protein, will also serve as its weakness, becoming the unique molecular identifier that our immune systems will remember to resist COVID-19.